video lecture we are talking about Southern blotting. What is Southern blotting? It is the analytical technique used in molecular biology, immunogenetics and other molecular methods to detect or identify DNA of interest from a mixture of DNA sample or a specific base sequence within a strand of DNA. This technique was developed by molecular biologist E.M. Southern in 1975 for analyzing the related genes in a DNA restriction fragment and hence named as Southern blotting. Now we are discuss about how Southern blotting gonna work. The first step is the extraction and purification of DNA from the cell. In this, DNA is first separated from the target cells following standard method of genomic DNA extraction and then purified. The second step is restriction digestion or DNA fragmentation. Restriction endonucleases are used to cut high molecular weight DNA strands into smaller fragments. One or more restriction enzymes can be used to achieve such fragments. Restriction enzymes are the enzymes which cut the DNA into small fragments. The third step is separation by electrophoresis. The separation may be done by agarose gel electrophoresis, in which the negatively charged DNA fragments move toward the positively charged anode. The distance moved depends upon its size. The DNA is extracted from somewhere, a genomic DNA of an animal. Then it undergo fragmentation by restriction endonuclease enzyme, and then it undergo agarose gel electrophoresis. Under the electrophoresis, the negatively charged DNA moves toward positively charged cathode. The fourth step is depurination. What is depurination? Depurination is a process in which adenine and guanine are separated from each other by breaking of N-glycosidic bond with the help of certain chemicals. Partial depurination is done by using a dilute chemical HCl, which promote higher efficiency transfer of DNA fragment by it breaking down into small pieces. The fifth step is denaturation. DNA is then denaturated with mild alkali such as alkaline solution of NaOH. This causes the double-stranded DNA to become a single-stranded and making them suitable for hybridization. DNA is then neutralized with NaCl to prevent rehybridization before adding of probe. The sixth step is blotting. What is blotting? Blotting is a technique in which transfer of some material from one place to another place. The denaturated fragments are then transferred on a nitrocellulose filter membrane, which is done by placing the gel on the top of the buffered saturated filter paper, then laying nitrocellulose filter membrane on the top of the gel, and finally some dry filter paper are placed on the top of the membrane. The fragments are then pulled toward the nitrocellulose filter membrane by capillary action and result in the connect print of the gel. You see in the diagram how blotting is done, and before blotting, depurination and denaturation is done. In depurination, adenine and guanine are separated from each other with the help of some chemical. In denaturation, the DNA fragments are separated from each other with the help of certain chemical like dilute HCl, and it prepare the DNA fragment for hybridization. And after that, NaOH also used, which does not undergo its rehybridization. In a blotting, DNA fragments are transferred on a nitrocellulose filter membrane, which is placed on the top of the gel. After that, some dry filter paper is placed on the top of the membrane. Finally, the membrane is pulled with the help of capillary action, and the result is print on that. The seventh step is baking. In baking step, the printed DNA on a nitrocellulose membrane is heated 2 to 3 hours at 80 degrees Celsius or exposed to ultraviolet radiation, which cause permanently attach the transferred DNA onto the membrane. Eighth step is hybridization. In hybridization, the membrane is exposed to hybridization probe. What is probe? Probe is a single DNA fragment with specific sequence whose presence in the target DNA is to be determined. Probe is labeled so that it can be detected by using radioactivity, or it can be tagging with the molecule with fluorescent or chromogenic dye. What are fluorescent and chromogenic dye? These dyes show some strange color apart from that molecule. Ninth step is washing. After hybridization, the membrane are thoroughly washed with buffer to remove the unbounded probe. 10th step is audioradiograph. The hybridized regions are detected with audioradiographically by placing the nitrocellulose membrane in contact with a photographic film which shows the hybridized DNA molecule. The pattern of hybridization is visualized on X-ray film by audioradiography. In case of radioactive or fluorescent probe, is used or by the development of color on the membrane if chromogenic detection method is used. After blotting step, the fragments are hybridized with phosphorus 32 probe. After probing, removing of unbounded probe with the help of certain buffer. After probing, removing of unbounded probe with the help of certain buffer. The last step is audioradiograph, which can be obtained by placing the membrane or exposed it to X-rays.